Welcome to BioRatna. Dear students, I am Chandana Sengupta and our today's topic of discussion is HGP. HGP, that is Human Genome Project. Students, I have already discussed half of the part in my previous lecture, Human Genome Project 1. If you have not gone through it, then please do. I can say this much that part 1 is fact-based, knowledge-based one, whereas you need to be more careful for this part 2 as it is concept-based, understanding-based. You need to understand the things, you need to be more careful. वैसे ये कोई बात नहीं हुई, हर चीज़ को आपको उतने ही फोकस से पढ़ना है, but you know what I mean. This is rather a that हिंदी में कहते हैं ना, सावधानी हटी दुर्घटना घटी, वैसा ही वाला ये टॉपिक है. तो अपना ध्यान बनाए रखें. So let us start with today's session. Students, when you started learning genetics in class 12, the two chapters of genetics, the principles of inheritance and variation and the molecular basis of inheritance, especially the second part, that is the molecular basis of inheritance, there you came across, you came to know, you understood the journey of the scientist, where first they were struggling to find out the structure of DNA, not knowing the structure of DNA. And here they have successfully sequenced all the nitrogen base pairs which are present in the half, that is haploid set, not half we cannot say, that is haploid set of chromosomes. Not just human being, many non-human organisms genome was also sequenced. What are they? If we see the animals, the 4 names which are given in your NCERT, they are bacteria, yeast, Drosophila and Caenorhabditis elegans. This Caenorhabditis elegans, it is a free-living, non-pathogenic nematode. Non-pathogenic nematode. Fine. This is about animals. What about plants? In case of plants, the genome of rice and Arabidopsis was sequenced. Why we took trouble to find out the genome of non-human organisms? To understand their natural capability. Here we are working for human welfare. We are trying to understand natural capability of these other organisms and so as to use it for various purposes. It can be used in healthcare. This information can be used in healthcare. It can be used in agriculture, or it can be used for energy production or environmental remediation. हम इसे कई तरह से इस इंफॉर्मेशन को हम दूसरों की ओर से आंख बंद करके सिर्फ अपना ही जीनोम देख के थोड़ी बैठ सकते हैं. We are studying the genome of other organisms also. We are finding out what are their natural capabilities and then we are using this information for our own welfare. And now we understand that it is not just the welfare of human being which matters. That is a different story. Now let us see the methodologies which were used for HGP. The methodology. The methodology for HGP. First, let us see the two different types of approaches. Students, while starting with the sequencing of the DNA, the scientists, they took two different approach. One group of scientists, they thought that, do you know that only 2% of the whole DNA, it expresses itself. It is genetically, it is transcriptionally active. Only 2% it is transcriptionally active. It is forming the proteins and it is controlling our traits. So, one group of scientists, they thought that what is the use of sequencing the rest 98%? Why to do that? They thought it as the wastage of energy, money and time. So they sequenced only the 2%. So what is the first approach? This first approach, which is called ESTS, here the scientists focused on identifying that part of DNA which expresses itself as what? As mRNA. Fine. So this full form of ESTS is the mRNA which is expressing. Let us tag only that. That's why this is expressed. This is expressed sequence tags. Express sequence tags. So they thought that sequencing of 98% rest of DNA is just wastage, but one wonderful treasure was hidden there, which has many different applications. Okay. So this was the first approach, and the next approach, this is called blind approach. Blind approach. In this blind approach, this scientist, they were going to sequence the complete DNA. In blind approach, the scientists were going to sequence 100% DNA. That means coding as well as non-coding. Complete DNA. Why is it called blind approach? Why it is called blind approach? See, in this approach, in the first approach, where the scientists, they have to code, they have to sequence only 2%, then first they should know which 2% is expressing itself as mRNA. You are understanding what I am saying? जब उन्हें सिर्फ 2% ही को कोड करना है, सीक्वेंस करना है, then they should know what is this 2% and what rest 98% is the non-coding part. तो पहले उन्हें देखना है, उसके बाद उन्हें काम शुरू करना है. That's why, and this approach, उन्हें तो वो देखने की जरूरत नहीं है, कौन सा 2%, कौन सा 98%, चलो पूरा का पूरा करते हैं. That's why this is called blind approach. So it is sequencing the 100% DNA. आपने पूरा कर लिया, and when the work is completed, after the work is completed, that after the completion of work, then also you have to tell which part is functioning for what, which is the coding part, what work it is doing. So after completion, the different parts parts are annotated with function. Annotated with function. That means इन्होंने पहले ही देख लिया कि किसका क्या फंक्शन है, बस उसी वाले को हम करेंगे सीक्वेंस. एंड हियर आफ्टर द वर्क इज कंप्लीटेड देन द डिफरेंट पार्ट is annotated with function, and this is called sequence annotation. This modus operandi is called sequence annotation. So these are the two different approaches. क्या approach लिया, अब उसका क्या steps है, let us see that. Now let us see the various steps of HGP, that is the process of HGP. What we are going to sequence? We want to sequence DNA, and DNA is present inside the cell. That's why the first step is isolation of DNA from the cell. हम उसे सेल से बाहर निकाल लेंगे. What is the second step? Now we make many fragments of DNA. Why we are going to do fragments of DNA? We were going to sequence DNA. तो अब उसे छोटे छोटे टुकड़े करने की क्या जरूरत पड़ गई? You know that DNA is a very long molecule and there is many technical difficulty in handling such a long molecule. That's why we make small fragments which are easy to handle, small fragments with the help of one enzyme. The second is making fragments of DNA with the help of the enzyme. The name of the enzyme is restriction enzyme, or you can say restriction endonuclease. Restriction enzyme or restriction endonuclease. Nuclease means which cut the nucleic acid. Endo means जो अंदर से काटता है. If this is the nucleus, if this is the DNA, then it will cut it from the inside, not from the sides. Fine. So it is called endonuclease. So making the fragments with restriction enzyme. Now that you have got the fragments, क्या अब उसका sequencing start कर दे? क्या उसके नाइट्रोजन बेस का सीक्वेंस खोजना शुरू कर दे? No, we don't do that. Why? आपके पास सारे फ्रैगमेंट्स के ओनली वन पीस है, सिंगल पीस है. So suppose during your work, during your experiment, during the work, somehow it is mishandled, then these steps you will have to repeat the steps again. But we don't want to repeat these steps. So what we do, once we get all the fragments, then we make multiple copies of the fragments. So next is to make the multiple copies of all the fragments which we have generated of the fragments. So how do we make these multiple copies? कैसे बनाते हैं इसको? We introduce it into one organism which reproduces very fast, and during that process it replicates its DNA. At the same time it will replicate our fragment also. Before explaining you this step, let me revise these steps again. First, DNA को निकालना था सेल से बाहर, निकाल लिया. बहुत बड़ा मॉलिक्यूल था, नॉट इजी टू हैंडल, फ्रैगमेंट्स बना लिए. कौन सा एंजाइम था? रेस्ट्रिक्शन एंजाइम और रेस्ट्रिक्शन एंडोन्यूक्लियस. अब हम नहीं चाहते कि एक सिंगल फ्रैगमेंट से अपना काम करे. We want multiple copies of each fragments. That's why now we are going to make multiple copies. How we do it? We introduce it into some organism. So we will call it host organism. We will introduce our gene into this organism which reproduces fast. So that it is reproducing fast, it is replicating its DNA and it will replicate our fragment also. And that DNA which helps our fragment, which this carries our fragment, which carries our fragment inside this host, it is called vector. So what is vector? Vector is the DNA which carries the foreign gene. Why our gene is called foreign gene? It is our gene, na? Why we call it foreign gene? Just think, I am telling you, but just think for 2 minutes. The DNA carries the DNA which carries the foreign gene into the host. It is called vector DNA. This fragments of ours is called foreign gene. It is called foreign gene because it is to be introduced into this host, and for this host it is a foreign DNA. There are 2 types of hosts which we normally use, that is, one is bacteria and one is yeast. If we are using bacteria, then the vector which we use is BAC. BAC means bacterial artificial chromosome. And if we are using yeast as host, these two they are host. Then the vector, it is YAC. It has to be chromosome. Here we have seen that vector is also a DNA. That DNA which will insert this our DNA fragment into it, and then it will enter into the host, and then our fragment gets multiplied. So it is BAC and YAC. These are the vectors. These are the vectors which help in the making multiple copies of our gene inside the host. Thus we get the multiple copies. Now what is remaining? Copy मिल गई, तो मेन काम शुरू किया जाए. अब तक सीक्वेंसिंग कहाँ स्टार्ट हुआ बच्चों? इतना सारा पढ़ते पढ़ते अगर भूल गए हो, what is the main work? That sequencing is to be started now. Now that we have obtained the multiple copies of the fragments, so we can safely start with our main work, that is to find out, this is the 4th step, na bacha? So this is to find out the nitrogen base sequence in the fragments. So how this work is done? You know, there was one scientist called Sanger. He was Frederick Sanger. What work one important he had done? He had found out a method to find out the sequence of amino acids in proteins. As DNA are made up of nucleotides and proteins are made up of amino acid, वहाँ का भी पूरा sequence amino acid का in proper order कैसे निकालना है, that was found out by Sanger. Now here, while sequencing of nitrogen base is done, this is done in automated DNA sequencer which works on the principle which was developed by Frederick Sanger. तो मिल गए आपको पूरे सीक्वेंस मिल गए, मेन काम आपका हो गया. अब एक बहुत बड़ा सा काम बाकी है. What is that? We have generated many fragments. So many fragments are there. Now we have to regenerate our chromosome, na? We have to regenerate the chromosome. Now we have to align all these fragments in proper order. That means alignment of fragments in proper order. This has to be done so as to regenerate this proper sequence in the chromosomes. We have found we are human beings, the most intelligent person on the earth. हमारे पास सारी चीज़ों के लिए इसके लिए भी एक प्रोसेस है कि how to after the fragments are made, then also we can now arrange it in proper sequence. This alignment, students, is humanly not possible. It is humanly not possible. So special computer programs were generated for the alignment of the fragments to regenerate the complete chromosome. And for this, the help of overlapping regions was taken. The help of let me write it here. And for this, the help of overlapping region was taken. Overlapping regions which were generated in the fragments. Overlapping means what? If this is one fragment and it is having here A, T, C, then this is another fragment which is having T, A, G. Can you see? One fragment, this is fragment 1. This is fragment 1 which is having A, T, C, and this is the fragment number 2 which is having T, A, G. So automatically these two will come together and they will join together. So with the help of the overlapping regions, the proper order, the alignment of the fragments, proper alignment of the fragments was done. तो बन गया आपका पूरा क्रोमोसोम. क्या आपका काम हो गया? Not now, not yet. Now you have sequenced नाइट्रोजन सीक्वेंस, बेस सीक्वेंस मिल गया आपको, आपने उसको प्रॉपर ऑर्डर में लगा दिया. अब नाउ यू हैव टू टेल व्हाट इज दैट 2% एंड विच वन इज द 98%. दैट मीन्स द 6th point is sequence annotation. Sequence annotation. The different regions in the sequence are annotated with a proper function. The sequence annotation is also done. So these are the different steps of HGP. Let me summarize it once more. पढ़ते समय तो बहुत सीरियसली 1 by 1 स्टेप से पढ़ना पड़ता है. अब हम लोग उसे ऐसे पढ़ेंगे. We have to sequence DNA. DNA is present in the cell. So first we have taken out the DNA. Second, DNA is a very long molecule, very difficult to handle. So we make it small fragments with the help of enzyme restriction endonuclease. बना लिए fragments. Third, now we do not want a single fragment. Single fragment, we want to have multiple copies of each fragment. So with the help of vector, we insert these fragments into the one host, which may be bacteria, which may be yeast, and which multiplies, and helps in the replication of or making multiple copies of our fragment. मिल गया fragment? Now we do that work for which we are here. That means we find out the nitrogen based sequence in each fragment. अब तो सारे इतने सारे fragments हैं, तो अब इन fragments को जोड़ना है. अब कैसे जोड़े इतने सारे हजारों fragments को? सो इन फ्रैगमेंट्स को हम जोड़ते हैं विद द हेल्प ऑफ ओवरलैपिंग रीजन्स. ओवरलैपिंग रीजन से भी जोड़ना इट इज़ नॉट ह्यूमनली पॉसिबल, सो कंप्यूटर प्रोग्राम्स वेर मेड फॉर दैट. Once the whole sequence in proper order alignment is done, then sequence annotation is done, that is assigning the different parts with proper function. Is this interesting or difficult? So these were the different steps in the sequencing of DNA. Now there was one more challenging task, that was to assign the genetic and physical map, on genome. And to do this, to assign the genetic and physical map on genome, we took help of two things. What was it? The first one, it is the polymorphism in the restriction site, restriction enzyme, in the restriction enzyme recognition site. एंजाइम रिकॉग्निशन साइट, क्या समझ में आया? Restriction enzymes you just studied, with the help of which we made the fragments of the DNA. So these restriction enzymes, wherever they are cutting the DNA, that is the recognition site of DNA for the restriction site. उसे भी पता है कि ये मुझे यहाँ पे काटने वाला है. So polymorphism of that site, polymorphism in restriction enzyme recognition site. This is one thing. And the second thing is some repetitive DNA called microsatellite. This is microsatellite. And you know the irony, students, this repetitive DNA, this microsatellite, it is the part of that non-coding part of DNA, that heterochromatin, for which it was thought that why is it necessary to code this 98% DNA which is not coding for any proteins. And we found this microsatellite in it. And this microsatellite, one application is to assign the genetic and physical map. One more application of repetitive DNA is in DNA fingerprinting. DNA fingerprinting तो हम कर ही नहीं पाते अगर ये 98% की खोज हमने sequencing अगर हमने नहीं की होती. But there we are not using microsatellite. There we are using something different, that is not mega. That is mini satellite. There we use mini satellite in case of DNA fingerprinting, and we do DNA fingerprinting with the help of mini satellite. Now let us see the salient features of human genome. Now let us see the salient features of human genome. Salient features of human genome. यानी हमने जो ये DNA sequencing किया, by that we came to know many properties of the genome. खजाना लगा ना हमारे हाथ में. So what is that? The first feature is that the genome, it consists of 3164.7 million nucleotide bases. Fine. कितने बेस है पता चल गया? Now, and every gene is made up of around 3000 nitrogen bases. It is made up of about 3000 nitrogen bases. थोड़ा कम थोड़ा ज़्यादा, but approximately 3000 nitrogen bases. But exception is dystrophin. It is how long? It is so very long that it is 2.4 million bases it is having. जस्ट सी, कहाँ 3000 और कहाँ 2.4 million. Now the third, the genes present in human genome, they are approximately 30,000 in number. Previously it was thought to be 80,000 or 1,40,000. Then those genes which we have discovered, from the discovered genes, we do not know the function of, 50% of them. 50%, इतना काम आप लोगों के करने के लिए बाकी है अभी. 50% of them. Okay, now the fifth one, only 2% of genome, it forms protein. Means it is the coding part. Sixth, 99.9% nucleotide bases are same in all human being. In all. तो इतना फर्क क्यों? Still you see ना, even if 0.1% is so much more, it is 3164.7 million nitrogen bases are there. So that 0.1 also is going to make huge difference. Now listen carefully. Now we see what is repetitive DNA. What is repetitive DNA? After that we will see what is SNPs. SNPs, pronounced as SNPs. First, repetitive DNA. This repetitive DNA is a repeated sequence of nucleotides. कोई एक न्यूक्लियोटाइड सीक्वेंस, कोई एक नाइट्रोजन बेस्ड सीक्वेंस इज रिपीटेड मेनी टाइम्स. आई रिपीट अगेन, कुछ नहीं, सिंपल है. कोई भी एक सीक्वेंस ले लो, आप कोई एक सीक्वेंस ले लो, AGCACT, ठीक है. Now this sequence will be repeated many times, like it will be again AGCACTA and so on. So many times, means this sequence may be repeated 100 or even 1000 times. When such repetitive parts they are present in DNA, then these do not code. These do not code for any protein. They are non-coding part. Not coding for any enzymes. So what is their work? Don't they have any function? They have some applications. Function also. They help in chromosome structure and dynamics, and they also help to understand evolution. How? Means we can trace back, means from where our ancestors have come and where they got settled. Every race can find this out with the help of repetitive DNA. So I repeat about repetitive DNA. Repetitive DNA, that part of DNA where a particular nitrogen based sequence, यहाँ से यहाँ तक लिया, then this will be repeated again, then again, maybe 100 or 1000 times, and it does not code for any protein, but it has this function. Then now the last, that is SNPs. What is SNPs? Here the S, it stands for single. N stands for nucleotide. And this P stands for polymorphism. नाम बड़ा बड़ा है बच्चों, है बहुत सिंपल. सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म. आपको लगता है बहुत मुश्किल है. सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म, नहीं उसको समझाने से अच्छा एक एग्जाम्पल दे दे तुरंत, आपको उस एग्जाम्पल से आपको समझ में आ जाएगा. देन आई विल गिव यू द डेफिनेशन. While studying sickle cell anemia, you have studied sickle cell anemia. While studying sickle cell anemia, you have studied that when in template DNA the sequence is CTC. This is the normal one, CTC. If it changes to CAC, this CTC, it was giving rise to normal hemoglobin. This was normal hemoglobin which we all are having. एक सिंगल न्यूक्लियोटाइड, हियर जस्ट एक सिंगल न्यूक्लियोटाइड में चेंज हो जाने की वजह से मॉर्फिज्म हो गया, पॉलीमॉर्फिज्म. मीन्स पहले सारे के सारे 100% नॉर्मल हीमोग्लोबिन वाले थे. When there was mutation and then this T changed to A, then this person become sickle cell, having sickle cell hemoglobin. दो तरह के मॉर्फ्स हो गए, पॉलीमॉर्फिज्म हो गया. क्यों? बिकॉज ऑफ सिंगल न्यूक्लियोटाइड चेंज. दिस इज द सिंगल न्यूक्लियोटाइड चेंज. सो लेट अस डिफाइन सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म. Single nucleotide polymorphism is the change in single nitrogen base in a gene which gives rise to different characters. Fine. One was normal hemoglobin, one was sickle cell anemia. How many means the scientists have found out about 1.4 million जगह. साइंटिस्ट ने इतनी सारी जगह खोज के निकाली है हमारे डीएनए में जहां एक पर्सन से दूसरे पर्सन का सिर्फ एक नाइट्रोजन बेस वैरी करने की वजह से डिफरेंस ऑफ कैरेक्टर हो गया है. आई रिपीट अगेन, ड्यू टू ओनली सिंगल नाइट्रोजन बेस वेरिएंस द पॉलीमॉर्फिज्म हैज ऑकर्ड. सच 1.4 million sites have been discovered. सो इससे हमें बहुत मदद मिलने वाली है इन बीमारियों को समझने में, जानने में, ट्रीटमेंट करने में. द स्टूडेंट्स, द HGP has given us much large many information, but the vast information which it has generated, अभी उसको और एनालाइज करना है और अच्छे से समझना है, so that we can understand our biological system properly. तो अभी और बाकी है, करना है, आप ही लोगों को करना है. अभी और उसको समझना है, जानना है, करना है. जैसे अभी तक के साइंटिस्ट व्हाट दे वर डूइंग, पहले जब जीनोम प्रोजेक्ट नहीं हुआ था, दे वर वर्किंग ऑन सम 1 जीन और 2 जीन. नाउ ऑल द जीन्स आर अवेलेबल टू देम. ऑल मीन्स जीन्स के फंक्शन पता नहीं 50% के, बट वी आर नोइंग 50% जीन, देन देयर mRNA, their protein, everything is known to us. So we can work upon कि जैसे कोई one tissue or organ or tumor, we can study the complete transcripts, all the transcripts in them. या फिर हम ये काम कर सकते हैं कि how this hundreds and thousands of the genes, they are working together like an orchestra. ना ऑर्केस्ट्रा में जैसे अलग अलग इंस्ट्रूमेंट्स होते हैं और टुगेदर दे फॉर्म सच अ ब्यूटीफुल म्यूजिक. सिमिलरली हाउ दिस tens thousands of genes, they are working together and orchestrating this beautiful tune, this chemistry of our life. ये काम रहा आप लोगों के लिए. बाकी काम, यहाँ तक का काम जो साइंटिस्ट ने कर दिया है वो हमने आपको समझा दिया. Hope the video was useful to you. Stay connected, stay tuned, do subscribe. Thank you for watching the video.